Good morning. It's January 8th, and this is your daily brief in science. Here's everything you need to know. A new set of hominin fossils from Grotta hominids in southwestern Casablanca, Morocco, dated to about 773,000 years ago, suggests an African origin for early Homo sapiens lineages during a key divergence period. The remains show a mosaic of primitive and evolved traits, lacking a defined chin, but with teeth and dental features similar to both Homo sapiens and Neanderthals, indicating a close yet not fully modern ancestry. Access to the full article is restricted to subscribers, with the complete text requiring login. The site indicates a dangerous environment with carnivore activity, including hyenas, reflected by bite marks on a leg bone and other signs of predator presence in the cave. Among the most informative remains are a nearly complete child mandible and 2 adult jaw bones, with one mandible more robust, suggesting possible sex differences. Most fossils were excavated in 2008 to 2009 but dated recently, aligning the layer to a major polar reversal to anchor the specimens in a defined time window. The researchers emphasized the importance of African fossils for understanding human evolution, and acknowledge ongoing uncertainty about the precise nature and identity of the last common ancestor of major human lineages. The article describes hominin evidence from Thurs-GH that includes a gracile adult mandible, a larger mandible, vertebrae, and teeth, indicating a small-bodied individual with a mix of archaic and derived features. Questions remain about whether these hominins were predators or victims of scavenging, with the cause of death not determinable from available evidence. The study highlights ongoing relevance of science journalism and the need for financial support to sustain science coverage. Overall, the findings contribute to the debate on African origins and the complexity of hominin evolution during this critical period. A breakthrough in nerve health shows that transferring mitochondria from glial cells to sensory neurons can ease pain and potentially repair small nerve branches in diabetes models. Dorsal root ganglion glial cells move their mitochondria to neurons through tunneling nanotubes, and stopping this transfer speeds up nerve degeneration and pain. The process uses TNTs, endocytosis, and gap junctions, blocking agents like Cyto-B, Y27632, Pitstop2, and carbonoxolone reduce transfer and TNT formation, revealing multiple coordinated mechanisms. Human relevance is supported by observing TNT-like structures between human SGCs and neurons, and snRNA-seq indicates SGCs are a major DRG cell type with conserved markers, underscoring translational potential. Lineage tracing supports unidirectional transfer from SGCs to neurons, with neuron to SGC or macrophage to neuron transfers appearing minimal under tested conditions. This mitochondrial exchange aligns with broader evidence that intercellular mitochondrial transfer supports health in obesity, cancer, stroke, and chronic pain contexts. In live mice, about 23% of sensory neurons contain glial-derived mitochondria after labeling, with transfer depending on endocytosis and TNTs. Mitotag in vivo data show time-dependent transfer increasing by day 10, and pharmacological blockers reduce transfer, reinforcing TNT and endocytosis roles in living tissue. Therapeutic implications include targeting TNT pathways, or MYO10, to modulate SGC to neuron mitochondrial transfer for pain management and CIPN treatment. Chemotherapy-induced neuropathy and stress reduce TNT-LS and impair SGC to neuron transfer, linking this mechanism to pathology and potential interventions. Transferred mitochondria from SGCs help reduce neuronal hyperactivity and ROS after injury or paclitaxel exposure, contributing to neuronal protection and possibly influencing small fiber neuropathy. Mitochondrial transfer is activity-dependent and increases with neuronal hyperactivity or nerve injury, suggesting a protective response in certain neuron subtypes. A new Chandra video tracks Kepler's supernova remnants' 25-year evolution, compressing data from 2000, 2004, 2006, 2014, and 2025 into a 40-second time-lapse that shows the remnant expanding into space. The project uses X-ray observations to illustrate the afterlife of the Type Ia explosion seen in 1604 by Johannes Kepler. Analysts examine the blast wave rims to understand the explosion and the surrounding medium, revealing how the remnant interacts with its environment. By studying the blast wave rims, researchers infer the nature of the ambient material into which the progenitor exploded and how the remnant evolves within that context. Jesse Gassel of George Mason University led the team, presenting the findings at the American Astronomical Society meeting in Phoenix, with NASA Goddard Space Flight Center overseeing the Chandra mission and the Chandra X-ray Center managing operations. The study sits within ongoing NASA Chandra efforts, framing Kepler's remnant as a crucial window into how explosive stellar deaths shape their galactic surroundings. The Chandra data illustrate the interaction of the remnant with surrounding material and trace its continued expansion over time. Gassel noted that the sequential data allow near real-time watching of the remnant's evolution over centuries. Researchers emphasized how the remnant's interaction with ambient material informs explosion dynamics and environments, as discussed at the AAS meeting in Phoenix. Kepler's supernova, observed in October 1604, became the brightest star in the sky and is tied to challenges to Aristotelian cosmology. X-ray observations show the remnant's debris glowing at millions of degrees, with Kepler located about 17,000 light-years away, enabling detailed time-resolved imaging. A new look at Brusselstown Ring in Northern Ireland is reshaping our view of prehistoric life in the region. Researchers at Queen's University Belfast combined existing terrestrial data, 288 hut site candidates, with aerial surveys revealing over 600 topographical anomalies, and added new excavations. They identify 98 potential roundhouse footprints within the inner enclosure and as many as 509 more between the inner and outer enclosures, suggesting a much larger and more complex settlement than previously thought. The project builds on prior findings to reassess occupation history and settlement structure, offering deeper insight into how people lived there. Dr. Dirk Brandherm notes the findings challenge established ideas about settlement organization, pointing to greater social complexity and regional significance. Even if not all anomalies prove to be roundhouses, Brusselstown Ring would still be the largest nucleated prehistoric settlement in Ireland and Britain by a wide margin. Nearby, a hill fort complex near Baltinglass in County Wicklow is identified as the largest nucleated settlement in prehistoric Ireland and Britain and may represent Ireland's earliest proto-town. Brandherm emphasizes that Brusselstown Ring would remain the largest nucleated settlement regardless of how many anomalies become roundhouses, with few other sites matching its footprint and not all enclosed. Fieldwork continues, with researchers stressing the site's national and international heritage importance and its relevance to understanding social complexity and regional patterns in prehistoric settlement. Ongoing excavations at Brusselstown Ring underscore its heritage significance and offer insight into community cohesion and regional organization in prehistory. Evidence points to a water cistern in a boat-shaped topographical feature, aligning with Iron and Bronze Age cisterns found elsewhere in Europe. The site features a necklace of up to 13 hilltop forts, 7 major hill forts, and associated enclosures dating from the early Neolithic to the late Bronze Age, roughly 3,700 to 800 BC. The research has been recognized with the Prehistoric Society's James Dyer Prize for 2025 and is published by Cambridge University Press, with funding from UKR1, NARC, and the Prehistoric Society. A new CRISPR tool could speed up rapid testing for multiple viruses by targeting RNA directly. In a landmark advance, researchers show Cas12a3 can be paired with other nucleases to detect influenza, RSV, and SARS-CoV-2 RNAs, paving the way for faster point-of-care diagnostics. The Utah State University team, led by Ryan Jackson with students Caden Crosby and Bamidele Filani, studies Cas12a2 and Cas12a3, which target RNA rather than DNA like Cas9. Cas12a3 uniquely cleaves the 3 tail of tRNAs, effectively halting viral protein production and enabling rapid pathogen-specific interference in protein synthesis for multi-pathogen detection. The study, conducted at Utah State University with international collaborators, was published in Nature and lists doctoral and master's students among the authors. The discovery grew from work at the Helmholtz Institute for RNA-based Infection Research and collaborators, with confirmation in Nature, and a focus on a distinct tRNA loading domain that positions the tRNA3 tail for cleavage. Funding comes from the R. Gorth Hansen family and the National Institutes of Health, with related patent activity noted in COI statements. Key collaborators span Europe and beyond, including several institutions in Europe and Austria, alongside institutes in Germany and Poland. Nature describes Cas12a3's ability to cleave tRNA tails to stop viral protein production while leaving host DNA intact, enabling targeted pathogen action. The Nature article is Oleg Dimitrenko et al. RNA-triggered Cas12a3 cleaves tRNA tails to execute bacterial immunity, with DOI 101038/S4-1586-025-09852-9. Cas12a3 uses an RNA guide to recognize foreign RNA and trigger a conformational change that enables targeted tRNA tail cleavage, offering a precise mode of action within the Cas12a family. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.